Okay. Thank you very much for the opportunity to be here. I'm very excited to talk about Elecular. I'm from Arizona State University, a professor in physics department. I'm the co-founder of Elecular. Elecular is a life science tool company providing cost-effective, scalable, single molecule detection with confidence for genomics, proteomics, metabolomics, and all aimed at realizing precision medicine. The promise of precision medicine really is based on digitalization of multi-omics. For example, without the help with next generation sequencer, we wouldn't be able to design a vaccine over the weekend. Without the accurate identification of proteins and metabolites, we would not be able to understand the molecular impact of an infection. All this data is the foundation for precision medicine and is a growing and large market. The workhorse for omics currently all comes down to two types of detection. One is the sequencer, the other is the mass spectroscopy. Both of these are bulk measurements that require a lot of samples, and also there are a lot of indirect sample preparation and translation of the information. A lot of important information is lost during that process, and also in many cases it provides a low throughput. We are starting to see the limitations of these technologies. For example, we need to detect a single cell out of 10 million cells in a blood sample to give us the clue of an impending cancer. Single cell and single molecule detection would play a more and more important role in the precision medicine. And this is the unmet market, and we want to be part of that revolution by replacing these instruments with a single chip platform. And we call this single chip the quantum nanoelectropore system. The unique part of this is that we're evaluating different intrinsic signals from a single molecule at the same time, all integrated in a single chip. How do we do that? Detecting a single molecule is very difficult, but in this single chip, we have many devices like this. Essentially, it has a single nanopore, which confines and detects the motion of and the position of a single molecule as we evaluate each one of them. And these pair of transverse electrodes give us the fluctuation of recognition tunneling current when they're very close to each other. That's tied to the structure of the molecule. In addition, this junction also enables the local enhanced Raman scattering signal, which tells us about this chemical signature of the modification of the molecule. Combining all these three different signals, we can then use a multi-parameter analysis to confidently identify what is running through the nanopore. So this is a label-free technology that does not require amplification to detect single molecules. We base our technology on standard semiconductor technologies, so we can outsource this to build a capital-efficient business model. We have prototype devices with preliminary data that shows this works. We're building our business model on a service-based model so that we can probe the market, so that we can tune our application to specific applications. And we're ready for a seed financing so that we can build our first forward model. We have a small team, but they're experienced in science and technology and development. So in summary, we provide a unified single molecule platform for multi-omic analysis. Thank you.
Perfect. Thank you so much for that. So how does this compare to, say, Oxford Nanopore with what they have on the market, and in particular, what Illumina might or might not have rights over?
Exactly. So that's why a little technical in my presentation so we can understand this better. So most of these nanopore detections are based on a single kind of signal, which is the ionic current, which means when the molecule comes through it, you see the blockade, and that's one type of signal that tells the size, and you try to guess what's in it. So there's a certain limitation. First of all, that fluctuation has a spatial resolution limited tied to the diameter of the pore. So the pore has to allow the molecule to pass, so you can understand there's a lower limit. So it's not a single base resolution intrinsically. And now for the DNA, it's easier because you only have 4 combinations. So you can guess basically 4 or 5 bases from that data. But when it comes to protein or RNA with modifications, the combination would explode, and there is no way you can solve that. So what we're doing here is using other intrinsic signals that's tied directly to the chemical structures. In this case, we have a tunneling electrode that is known to give a characteristic fluctuations when you have a molecule sandwich between two electrodes that's right next to it. And also we have the Raman signal that's going to be enhanced by this nanojunction. That's going to be also characteristic to the chemical modifications of the molecule. Combining these two, then you have a more confident call of the structure of the molecule instead of just guessing by the size of it. I hope that makes sense.
Thank you for that presentation. It was well done. Can you explain how what IP you have on this and what's shared with the university?
Yes, we did this. So our company was funded by, the research was funded by government money, as well as my startup in the ASU. So we filed the IP through the ASU, and we're in the licensing process after we found the Elecular in 2019. And that's where we are. We're in the licensing process.
One more question about manufacturing. So what kind of scaling plan do you have? Because my day job is in the contract manufacturing space, and I can tell you electronics, chips, and all of that's really tough right now.
As you can see, we already proved that this can work on wafer scale, and this is a 3-inch wafer, and this can scale up like any other semiconductor technologies, and there's nothing non-standard in the fabrication process. The real core technology is in fact not the chip, because the chip is really cheap to make. The finalized how we actually finish this pore is all contained in this electronic control system, because we're not fabricating the nanopore directly. Most of the other technologies, they use drilling process to finish the pore, which is irreproducible, and all the devices can perform differently. In our case, the pore is finalized during the recording process. So the electronic system actually have a feedback system to actually tune reversibly the size and the shape of the pore. So this is a multiplex system that you can tune every device to make it work. We have only a few devices on this single chip, but for the next generation, we hope to integrate thousands of devices in this single chip. So that, of course, requires additional development of the electronic system, but it's possible because there have been many examples of multiplex system developed in this way, and we just need to develop the software to support that.
You were talking about the need to sequence sometimes like a single cell out of 10,000.
Yeah.
So what technology do you see this partnering with to identify that one cell of interest from the milieu of the rest?
Yes, so we hope that our system can be combined with single cell analysis system. For example, you can have a single cell delivery lysis process ahead of this detection system, so that the content of the single cell is then multiplexed and then analyzed by a single chip. We basically estimate if you have, let's say, 100 microliter of content that you need to screen, you certainly, and especially for a single molecule detection system, most of the limitations of other technologies that you're limited not by the technology, but limited by the diffusion process. You have to wait very long for the molecule to pass, and you'd see that rare event you want to see. In our case, the channel that these devices are embedded in has a gradient of electric field that can increase the capture rate significantly for each one of these devices. And we estimate that for 100 microliter of material that you need to evaluate, we need to integrate about 100,000 devices. So then you can screen all of these contents with 95% confidence within, it depends on what you do, 1 to 10 hours. I think that's achievable target, because with the new generation of design we're working on, each of the devices occupy only about 10 to 30 micrometers square area. So that can easily scale up if we can have an on-chip preamplifier, multiplexers, and all these standard semiconductor structures in there.
Sorry, great, very interesting technology. I'm wondering in terms of the business model, are you planning to sell the devices?
Yes.
Or
Sorry, a
analyze
little
samples?
rush on this. We would like to construct the system and we supply the consumable chips. We have standard versions of the chip, so then it's for generic screening of single molecules. But these electrosurface, they are metals that can be modified chemically. So you can target specific biomarkers if you want to be very specific, so you can increase the contrast or specificity of detection. Therefore, we can have customization of that solution to different users. And that's the reason we want to provide a service, a subscription-based service to the users, so we can actually understand what is the need for different applications and can then find a new market for the product. We actually have a first alpha test user who's working with us to find the application of this in cancer therapy. They want to evaluate different biomarkers to evaluate the risk of a patient in their treatment in a very timely manner.
Nicely done, nice work. I had a quick question regarding your multiplexing. Did I hear you correctly that you said you can multiplex up to 1,000 analytes on a single chip?
Yes.
Okay.
Yes, so because these, so these chips now is, and this first generation, as you can see, is actually limited by the way we deliver the sample because everything is on one single surface. So you have to direct the different samples in different positions of the chip. But in the second generation, we actually use both surface of the wafer. Therefore, you can just segment the chip where then different samples can go through it. And for each segment, you can have many devices working together so that you can collect enough statistical data to evaluate that sample. So we believe there is a high potential to do that.
And do you foresee these chips to be reusable?
We hope so, because that needs to be evaluated. That if you are just doing generic detection, I think it's possible. But if you're looking, for example, a specific biomarker that binds or unbinds, that surface may get less and less effective as you use the chip. Thank you.